So it's my great pleasure to be introducing this year's winner of the Elizabeth D. Hay Award. Since 2017, the SDB has made this award to an investigator who's early in their independent career, performing original, significant research with impact on the broader field of developmental biology. Betty Hay, who gives her name to this award, was a woman of many firsts. In 1973, she was the first woman to be elected president of the SDB. She was subsequently the first woman to be elected president of the American Society of Cell Biology and the first woman to receive SDB's Conklin Medal. Betty Hay was also the first investigator to show that the extracellular matrix plays a vital role in determining cell behaviors. She's best known for her research in limb regeneration, the role of the ECM, and epithelial to mesenchymal transitions, and we give this award in her honor. This year's winner is Dr. Hernan Garcia, assistant professor at the University of California, Berkeley, who not only fills all the expectations of the Hay Award, but also personifies pretty much all of the values that we in the Society for Developmental Biology hold dear. I've learned that Hernan grew up in Argentina reading Isaac Asimov novels, which he credits with setting him on the route to becoming a physics student at the University of Buenos Aires. By 2003, when Hernan received his licenciado degree, Argentina was going through incredible political upheaval, and like many other young Argentinian scientists, he decided to get out of the country. Now, fortunately for us, he ended up as a PhD student at Caltech, and there he started getting interested in biology when he took a class in physical biology with Rob Phillips, whose lab he later joined. Now, during a spectacularly productive PhD, Hernan made and experimentally tested statistical models of the mechanics of bacterial transcription. But importantly, he also went to Woods Hole for the MBL physiology course, which was, in his own words, completely career-changing. It was there, visiting buddies in the lab downstairs who were taking the embryology course, that he first encountered fruit flies. The joint course speakers included Eric Wieschaus, and although Hernan was working on bacteria at the time, he saw the Drosophila work and thought to himself, quote, "This is quite a cool challenge." It was that experience, as well as a lunch conversation with Eric in the Swope dining hall about the methodological limitations to understanding transcription, that helped to determine Hernan's next career step, which included becoming a postdoc at Princeton with Thomas Gregor. While at Princeton, Hernan made amazing progress developing a system for precisely measuring the dynamics of transcription in the early fly embryo. And this led him to generate the first dynamical measurements of transcription in single cells within a living multicellular organism. These approaches led to several key insights, including the occurrence of transcriptional bursting, and the technology has been used by many other labs to answer a wide array of questions, thus having a significant impact on the field. As an independent investigator at Berkeley, Hernan has extended these methods to visualize both nascent transcripts and encoded proteins in living embryos. His remarkable findings include the visualization of waves of protein synthesis that are synchronized with the underlying transcriptional bursts. He's also gone on to apply these methods to the systematic dissection of defined transcriptional enhancers, and it's these efforts that provide the foundation for his long-term goal of unraveling regulatory codes across multiple organisms. Now, beyond his outstanding research contributions, which have already garnered multiple awards, Hernan's nominations for this award also highlight that he's a true scholar and an incredibly generous spirit, someone who's even willing to help competitors, because for him, the science always comes first. He also greatly values the training and mentoring of young scientists in his lab, and he loves teaching. He co-directs his own MBL course now, Physical Biology of the Cell, to inspire a new generation of scientists. Hernan commented to me that for Betty Hay, it was seeing electron microscopy images that got her hooked for life and redirected her career, while for him, it was seeing embryos. He also commented that the fact SDB is giving this award to a physicist says something good about our community. Well, I think what it really says is that we in the Society for Developmental Biology know a stellar developmental biologist when we see one. So please join me in congratulating Hernan Garcia on his award and welcoming him to talk.
Hello, everybody. It's such an honor to be here today. I just have no words for how humbling it is to be in front of you, all of you. I'm so grateful to all my mentors that helped me along the way, and most importantly to all the students and postdocs that did the hard work that I'm going to talk to you about today. And what I thought I would do today is basically give you what I would call my love letter to developmental biology or quantitative developmental biology. And it's fun, Vicky already gave some of my first slides. But after I talked to her, I started thinking about how as teachers, mentors, as communicators, we have the power to define careers with a single interaction. And I was amused to read this quote by Betty Hay, where she saw a talk by Keith Porter using EM for cell biology, and all of a sudden, it was all EM for her. And as Vicky mentioned, I was taking the physiology course in Woods Hole back in 2005, and it was pretty cool that they had this joint symposium with embryology, and Eric Bischoff came to give a talk about the work they were doing on the dynamics of formation of morphogen gradients in the fly. And I just couldn't believe that you could see these single cells as they were making decisions inside a living embryo. And I thought, I want to get a piece of the action. And at that point, I was a grad student at Caltech in physics. I went thinking I was going to do string theory or quantum computing, but like many in my generation, I did the jump from physics to biology. And I'm sure you know many examples like that, but probably there are not that many examples going from biology to physics. And and the cynical view would be to say, well, biology is easier. But my view is that there's just a lot more exciting stuff going on this side of the equation. It's one of the greatest mysteries in nature. So. I guess. So I'm super excited to share my love of math and physics to try to shed light on embryonic development. And so in my lab, we ask a question of how you go from a single cell to a multicellular organism. That's pretty familiar to all of you. And we do this for the most part in the fruit fly. And you've seen movies like this again and again. So you're looking at an embryo. The cells are going through multiple runs of synchronized division. And what I find fascinating is that at this point, all these cells look exactly the same to you. But as you know, they already know what they're going to be when they grow up because they're expressing a unique combination of transcription factors. And another thing we know, which is an amazing achievement of developmental biology, is the fact that the transcription factors that endow cells with their identity are organized in a network in the form of a cascade where you have patterns with increasing sharpness, right? From shallow gradients to broad domains to 7 stripes to a segmented embryo. And we don't only know the identities of these transcription factors, we also know the regulatory connections. So this is mind-blowing that we can do this. And this is not obviously unique to the fruit fly. From fruit flies to sea urchins to hematopoiesis, we're pretty good at mapping gene regulatory networks. And yet, despite the state of the art, I have a complaint. And that complaint is that just like this is not a pipe, this is not a fly. Okay? What I mean by that is if I change the names here and I give it to the world's experts on flies that are sitting here, they would have a hard time, I would have a hard time telling you that this is a fly. You cannot calculate a fly out of this. And what I hope to convince you of today is that the reason you cannot do it is because this description is missing information about space, about time, about concentrations, about the effectors, about allosteric interactions. So what we try to do in my lab is to endow each one of these arrows with quantitative and molecular information that allows us to reach a predictive understanding of developmental decision making. Basically, could I predict quantitatively the developmental phenotype from knowledge of the network topology? Okay? And we're not going to do this for the whole fly. We're going to try to focus on individual arrows in the beginning. And we're going to use a very useful abstraction, which is to think of these arrows as input-output functions. Okay? So how does the DNA arrangement of binding sites dictate the functional relationship between the output level of gene expression and the input concentration of your transcription factor? And you can think of some quantitative phenotypes that appear here, such as the leakiness, the EC50, the slope, the saturation. And I want to be able to understand this so well that I can predict how these phenotypes change as I tune the number, the placement, and affinity of transcription factors on the DNA. All right? And how are we going to do this? Well, we're going to use the classic dialogue between theory and experiment that has been the hallmark of the physics approach over the last 500 years. What I mean by that is that we're going to grab these cartoon models that we're so used to drawing in biology, and we're going to turn them into precise mathematical statements that make predictions, for example, about the rate of RNA production as a function of the concentration of activator. And then we're going to go to the lab and measure the input concentration, measure the output transcription, and see whether the predictions make sense. And the point is that this is a dialogue. Like, theory is there to make predictions that are experimentally testable. Experiments are there to explicitly designed to test those predictions and to inform the next round of theoretical modeling. And even if you don't care about quantitatively predicting development, I mean, we don't all need to like the same things. I claim that this approach is useful in terms of gaining biological insight into mechanism. Because as this is very much inspired by this great article that I recommend to you by Joel Cohen entitled Mathematics is Biology's Next Microscope, Only Better. Biology is Mathematics Next Physics, Only Better. So I'm curious about the idea of using, by thought, using physics and theory, being able to see things that you couldn't see with even the fanciest super resolution microscope. And there are very good examples of this already in biology. Think, for example, of the Luria-Delbruck experiment. By looking at distributions of bacterial plaques on a viral plaques on a bacterial lawn, they could figure out the nature of mutations. Or think of kinetic proofreading, like where Hopfield and Nino realized that the error rate in translation was too low to be explained by a thermodynamic mechanism, and they figured out that there had to be energy being used to fuel error correction mechanisms. Okay? Now, of course, these examples are maybe more on the molecular side, and you might say, well, if I want to understand how a whole cell makes decisions, it's going to be a little trickier, it's more complex. And I agree, it's harder. But at least in the case where we went at it systematically for a period of almost 15 years, or maybe more 10 years, we managed to do this. So this is work that I did in my PhD with Rob Phillips at Caltech, where we were looking at bacteria trying to predict how the LAC repressor binds to the DNA and excludes RNA polymerase from the promoter. And the point is that we make these theoretical predictions for the level of gene expression as a function of the number of repressors, where each curve that you see here corresponds to, for example, a different binding energy for the repressor to the DNA, or a different relative number between copies of your reported gene and number of repressors. The point here is that all these curves existed before we took the data. And then we took the data. And what we showed is that to a large extent, you give me the DNA sequence and I can predict the input-output function with relatively high precision. Not only that, all these curves look very different. They don't feel quite like they came from the same model. And that's because we're not plotting the data against the right x-axis. If you plot it against an effective number of repressors, what you can show is that all the data collapses onto the same master curve, which tells you that we've uncovered the natural variable of the system. Now, one complaint would be, well, this is bacteria, and eukaryotic transcription is more complex. Animals are more complex than single cells, and I would agree 100% with that. But what we need to do is just think about what are the challenges and work systematically at solving them. So what are the main challenges that one can imagine? Well, first of all, if we want to predict input-output functions, we need to be able to measure them. Second of all, we know surprisingly little about what activators and repressors do when they bind to the DNA. And if I don't have the cartoon model, I cannot write the equations corresponding to that model. And finally, we need to figure out how to derive and test models of developmental or cellular decision-making. And these challenges basically represent the main three research threads in my lab that we've pursued over the last 7 years, where we're devoted to lighting up the processes of the central dogma in developmental biology, to using physics as a microscope for in vivo biochemistry, and to combine everything we learned in order to realize physical biology of developing embryos. And we've made great progress on all of these fronts. And today I'm just going to give you a few snippets of some of the successes and, most importantly, the failures that we've had along the way. So the rest of the talk is going to be divided in 3 short parts. First, I'm going to introduce you to the technology we developed in order to be able to measure these input-output functions in single cells of living embryos. Then I'm going to show you a few examples of successes and failures in the theory-experiment dialogue in quantitative developmental biology. And finally, I'm going to spend some time trying to think about the challenges that we could maybe agree as a field that could be interesting to pursue, given what we've learned so far. All right. So let me take a step back and just say that I don't want to imply that before we started working on flies, there were no theoretical models in developmental biology, right? I mean, you know very well, there were lots and lots of models. In particular, thinking of the fruit fly, there were all sorts of interesting models based on statistical mechanics, on non-equilibrium processes, dynamical systems. There was no lack of theory, there were no lack of predictions. But what we lacked were the technologies to be able to test those predictions. All those models predicted input-output functions that we couldn't measure, okay? And why is that? Well, you know, what would you do? Like if you want to measure the input concentration of activator, your first reaction would be, well, I'm going to fuse it to GFP, right? And why can't you do that? Well, you know, many years after I met Vishwas at that talk in Woods Hole, we were actually teaching together in the physiology course, and we were having lunch in Swope, and I asked him, why haven't I seen this? And what he reminded me of is that when GFP is produced, it's dark, and it takes on the order of 40 minutes in flies, in fish, in worms, and frogs for it to become fluorescent, for it to mature. And this wouldn't be a big deal if it wasn't for the fact that the activators and repressors we want to look at live for 10 minutes. So by the time the fluorescent turns on, the action is over, okay? So there are some exceptions to this. People have managed to look at, for example, maternal gradients or stereotypical oscillations like in somatogenesis, okay? But for the most part, the field was constrained to looking at capturing embryos at different time points in their development, killing them, staining them, and then examining fake movies where you morph through images of dead embryos, okay? So this is a great state of the art, but clearly you cannot get single-cell dynamical information from this technology, okay? So over the last 7 years, we've worked systematically to be able to measure these input-output functions. We've developed technology to measure the output rate of transcription, to measure the input concentration of activator, to even manipulate the input concentration of activator with light. And I'm not going to talk about all of these technologies, but I just want to give you a flavor of the type of data we get by showing you the technology that we use to measure the output rate of gene expression, okay? So this is work from my postdoc with Thomas Gregor at Princeton, and this technology was developed in parallel at Nathalie Dostatny's lab at the Curie Institute in France. And it's all based on a really cool invention by Rob Singer at Albert Einstein that we re-engineered so we could make it work in an animal. And the idea is the following. We're going to add a little sequence of the gene of interest that is called MS2 that when transcribed forms a loop. The loop is recognized by a binding protein fused to GFP such that every nucleus in your embryo will have a little spot of fluorescence, and the fluorescence of that spot will be proportional to how many polymerase molecules are actively transcribing the gene. So this is what it looks like when you put it all together. So in red you now see the DNA again, and in green you see the RNA signal. Hopefully it shows up okay. Okay, so we're looking at a stripe of expression, and you should see a broad domain. And that broad domain over developmental time is going to become narrower and narrower until you get a little stripe of about 6 microns in width. This is, as Vicky mentioned, this is the first time anybody could look at transcriptional dynamics of single cells in a living multicellular organism. And not only that, one of the cool things about this is that you're staring at an in vivo single molecule experiment because each one of these dots reports on the transcriptional decisions made by an individual DNA molecule in each one of these cells inside the embryo, okay? So the output is just one part of the puzzle. We've also developed strategies to measure the inputs. So once again, here I'm showing you the stripe as it goes from broad to narrow. And it turns out that it becomes narrow because there are repressors coming from each side. In particular, there's this repressor here called Krupel that we can visualize with some new probes that we call LAM-ATACs. The name doesn't matter, but the point is that now we can measure both inputs and outputs in the same cells. And we can get information such as what I'm showing you there in the plot, where you can see in red how as a function of time the output is bursting, going up and down. And then when the Krupel concentration crosses a threshold, shown in, the Krupel in green shows it crosses a threshold, the transcriptional activity just disappears. Okay? So now we're ready to actually be able to measure these input-output functions to test our models. But these technologies, besides our specific research objectives, these technologies have slowly been becoming one of some of the standards in the quantitative dissection of developmental programs. And many labs are using it already in the fly and also in different organisms, often in the context of collaborations with us, which is a fantastic opportunity for us to learn more about other model systems. And I don't say this to boast. I say this because just to show that we're committed to nurturing a vibrant community of quantitative developmental biologists. And in fact, we have visitors all the time in my lab that are interested in learning how to implement their technologies in their systems or learning how to analyze the data. So if this is something that any of you is interested, find me during the rest of the meeting or just shoot me a message and we can find a time to talk. All right, so I showed you how we light up the central dogma by going beyond these static movies of dead embryos and actually putting real-world dynamics under the microscope. And now what I want to show you is how we use these technologies to engage in this dialogue between theory and experiment that I introduced you to at the beginning of the talk. And one example that I'm pretty excited about has to do with the fact that even though I've been drawing DNA as a linear molecule, we know very well that it doesn't look like that inside the cell. It's wrapped around nucleosomes. And we know that transcription factors that are wrapped around, that DNA that is wrapped around a nucleosome is not accessible by transcription factors, okay? So people like Oleg Igoshin, Rob Phillips, and Leonid Murney took this model seriously and turned it into math, okay? And the model basically tells you that DNA is gonna unwrap and wrap as a result of thermal fluctuations. And then when it's open, transcription factors can use that chance to bind to the DNA, okay? So we decided to test this model in the context of how Bicoid activates Hunchback. And one really cool prediction about the model is that if you measure the time it takes from mitosis for the spots to appear, for transcription to begin, that time should be constant as you move along the embryo. And the cool thing is that we can make those measurements very easily, like what I'm showing you here. So we can look at when the spot turns on and I can color the nucleus accordingly, okay? And we can compare theory and experiment. And this is what we got. Basically, there was no amount of parameter tinkering within this model that could match theory and experiment, okay? So the model failed. But what does this failure mean, okay? And I think we need to remember, for example, the great words in this article by Jeremy Gonawardena, that models in biology are not accurate descriptions of nature, but accurate descriptions of our pathetic thinking. And what he means by that is that when we build a model, it's based on assumptions, right? We build a cartoon model, we make some assumptions, we're baking in some assumptions in the drawing, and then we try to translate those assumptions into mathematical form. So what we're doing by confronting theory and experiment is testing the assumptions, our assumptions of how reality works, which means that we need to revisit our assumptions, okay? So what did we decide to do? Well, we thought, well, maybe transcription factors are not passive bystanders in this chromatin opening and closing game. In fact, there's evidence that transcription factors could spend energy to actively catalyze the opening of chromatin. And imagine that they could do it in an irreversible fashion, such that the DNA is now accessible and any transcription factor can bind there. This sounds a little more complex in terms of the math, but actually it has the same number of free parameters. And without much effort, you can show that you can recapitulate the experimental data. Now, I'm not claiming that this is the right model, okay? So obviously we need to find better new predictions to make. But what I'm trying to emphasize is that if you have enough confidence in your precision measurements and your theoretical models, a difference in timing of transcription as short as 4 minutes can shed light onto the dynamics of chromatin accessibility on spatiotemporal timescales that you can't access with any genome-wide approaches that I know of. And this is just one of many adventures. For example, another thing that we were interested in is thinking about shadow enhancers or sibling enhancers that talk to the same promoter. And the null model that you would imagine is one where they work additively, okay? So how do you test this? Well, you measure the transcription coming out of the case where you have both enhancers compared to the case where you have each enhancer independently and when you add up the activity. And we measured this quantity for a bunch of genes. And what we found is that indeed there's a regime where enhancers do work additively, which corresponds basically to a case where the enhancers don't interact very often with the promoter, so they don't see each other. We also found that there's a regime where the enhancers work subadditively, which corresponds to cases where the enhancer interacts so strongly with the promoter that it excludes the other enhancer from there. But surprisingly, we also found that enhancers can work synergistically, where they seem to help each other to activate the promoter. Another phenomenon that we've been very interested in is that of transcriptional bursting, where the promoter fluctuates stochastically between an on and an off state. And the simplest model that you can come up with is one that says that when the transcription, let's say an activator, binds to the DNA, the promoter switches on, okay? And a model like this makes very concrete predictions about the distribution of binding times that you should observe. So we decided to measure these binding types using lattice light chain microscopy. And what we showed, once again, is that there was a complete disagreement between theory and experiment. And as we examined the data more, what we discovered is that the fly nucleus is not a bag of well-mixed transcription factors, that this activator had increased binding frequency in certain discrete hubs throughout the embryo. And if you account for the presence of these hubs, now you can recapitulate the experimental data, okay? All right, so these are just one of many adventures we've had at the interface between theory and experiment. So I gave you 3 examples. We also discovered that enhancers can regulate the probability of a promoter turning on, and once the promoter is on, they can also dictate the rate at which they'll produce RNA. We showed that a simple model where you have glue-like buttons along the genome, they can predict the dynamics of somatic pairing in the fly. We also showed that all 7 stripes have even skipped, even though they're generated by different enhancers. All these stripes are sculpted through the same quantitative regulation of transcriptional bursting. They all increase burst frequency in the same way, for example. All right, so I hope I made it clear, hopefully I made it clear, that as much as we love our successes, we actually learn more from our failures. Okay, and I just want to emphasize that this is not a failure of the quantitative developmental biologists. This is a failure of all of us as a field, and I don't mean it in a critical way. I mean it in the fact that we have assumptions that we typically encode in the language of cartoons, that what we do is we try to take them seriously and turn them into mathematical equations. And the claim is that when you confront theory and experiment in this way, you sharpen your predictions, you become more sensitive to surprises, and you learn things that typically you couldn't learn with a more qualitative approach. All right, so now that I've given you a little bit of an idea of this theory-experiment dialogue and what we can do, I just wanted to spend the last few minutes reflecting on the challenges ahead for the field of quantitative developmental biology, or at least the challenges that I'm very excited about pursuing. So you might be surprised to know that even in E. coli, which arguably is the most understood organism on Earth, we only know the regulation of 33% of operons. For the rest of the operons in E. coli, we have no regulatory information whatsoever. Okay, we don't know anything about binding sites, we don't know nothing. And as you can imagine, the situation is worse in animals. Okay, if you look at databases, we only have regulatory information for 5% of the genes. So, when we have this picture of, check out how well we have mapped the even skip enhancers, this is just 5%, we have that sort of information for only 5% of genes, which means that all the approaches that I introduce you to can only be applied to that 5% of genes. Okay, so, and why is this? Well, it's hard to actually find binding sites. It's hard to do this. It's a one by one effort. So what I'm curious about is whether we can leverage what we have learned now about in vivo gene editing and high throughput sequencing to go from a state of ignorance about an enhancer to knowing where the binding sites are, who is binding there, to knowing their binding energetics, and being able to predict their input-output functions. So that's one of the challenges we're actually taking on in my lab that I'm very, very excited about. The other point I would like to make is that, despite my obsession with pattern formation, clearly making a gene pattern is not enough, right? The embryo needs to do stuff. You need morphogenesis ultimately. And I think it's such a fun challenge to try to figure out how you encode for shape, for forces within gene patterns. And you heard a bunch of interesting things about it during the conference already. And so I think there's something very interesting in trying to uncover the mechanical code in morphogenesis. Not only that, also from the physics perspective, this is a very interesting material. This is like an active material. And something very unique about it is that you have these molecular motors that are injecting energy into the system at the molecular scale. And somehow they need to work out how to encode for structure at the macroscopic scale. So it's a mind-blowing problem, I think, that is going to be really fun to try to solve. All right, so the last point I want to make is one that I realized when I was at a faculty meeting in physics where we were discussing infrastructure and how we're going to make the labs more temperature stable. And I started thinking, the most important infrastructure that nobody's talking about at that faculty meeting are the students and postdocs that actually do the stuff. And so this brought me back to this whole interesting history about Einstein when he was an undergrad at what later became ETH, where at the time, the coolest thing out there in research were Maxwell's equations of electromagnetism. And those equations were nowhere to be found in the classroom. Okay? So what I'm curious about is, are we really helping students become explorers and discoverers at the interface between physics and biology, between theory and experiment? Okay, are we really teaching, giving them the opportunity to thrive in biological numeracy in theoretical modeling? And one approach that we've been engaged with, especially together with Rob Phillips, is of teaching intensive boot camps at the interface between physics and biology, and very much inspired by the Woods Hole model. Like we love just owning people for 10 days and just going for it. And we do all sorts of interesting things. One cool experiment that we do is we try to test the French flag model. So we ask, if I was able to change the dosage of picoid, where would the new cephalic furrow be positioned as a function of the dosage concentration? And then we can use tabletop microscopes to actually do the measurements in the lab. This is an actual movie that students got in the lab at Berkeley, or got in the classroom, sorry, at Berkeley. And then they confront theory and experiment. And typically the reaction students get from this result is that they screwed up. Like they did something wrong with the experiment. And it's quite an insight to convince them that no, what you just found is that the model is wrong. That it's, the French flag concept might be right, but it's not true that picoid is the only determinant of where the cephalic furrow appears. And all the sorts of experiments that we do is they measure the elongation rate of RNA polymerase along the gene. We give them movies where we've labeled the beginning or the end of the gene, and they develop a data analysis pipeline to be able to measure when each plot appears. And when they subtract, when they get that delta t, they can figure out what the elongation rate is. All right. So I guess I'm just trying to finish on the note that I think this might be our most important contribution moving forward for the people that we mentor, that we help become better scientists. And speaking of that, I want to come back to thanking all the students in my group. These are the first student and the first postdoc in the lab. I was revisiting our pictures as time went on, and how it's just fun how the lab itself is a living organism, and it's just I just can't thank them enough for wanting to join me in this crazy adventure that we've embarked on. I also need to thank my official and unofficial mentors. I'm so lucky to have them around, and for them to, I know there's no way to pay any of them back, and I know it's cliche, but the only way to do this is to pay it forward. And especially I want to thank my physical biology of the cell family. These are my book co-authors, Rob Phillips, Julie Ferrio, Nigel Orme, Janne Condev, and Christina Hushen, who are really a scientific family that has taught me so much about science. And last but not least, my wife, who literally was supporting the family while I was hiding in the basement trying to finish papers so they could be ready for tenure. And yet now the final slide. I know we all hate writing grants, okay? And it's understandable, but I mean, just take a step back and think about the fact that they pay us to do this, right? So it's pretty crazy. I mean, it's such a privilege to be doing this. So I'm just so thankful to these funding sources for having given us the freedom to pursue the research questions that we're excited about. Again, very thankful to the Society for this award, and to all of you. And if you want to chat, I'll be around until the end of the meeting. Thank you very much.